So our next speaker is Kathy Nagel. Kathy was a graduate student with Allison and sort of also a graduate student with me in the sense that we co-published. And she went on to do a postdoc with Rachel Wilson and now has her own job at the medical school at NYU. Kathy.
Thanks, Steve. So I'm going to start with science and talk a little bit about Allison at the end. One of the things that was, I think, unique to Allison's scientific approach is that she was always very motivated by these questions of natural behavior, neuroethology, especially thinking about what are the actual stimuli that an animal is going to encounter in its environment as it's doing its thing. But she combined this with a very hard-nosed interest in mechanisms, which you've heard a little bit about already today, and also in using quantitative approaches and quantitative models to really get insight into what the nervous system was doing. And I think that that was the approach that drew me to her lab and that I tried to continue in my postdoc with Rachel, another UCSF alum, and now in my own lab. So I'm going to talk today about the olfactory system in the fruit fly, and I hope that you'll see what I learned from Allison and took into this system. So as I said, because of my training with Allison, when I went to study the olfactory system with Rachel, I started immediately thinking about what are the olfactory signals that a fly actually encounters as it's moving around its environment. And it's hard to measure odor signals in nature, but you can get an idea of what they do by looking at some other kind of tracer like smoke. So this is an example. If you have a source of smoke or a source of odor that's up in the air, it will tend to form what's called a plume, a turbulent plume. It kind of meanders its way downwind, but it has a very complicated structure that's hard to predict. And so if you're a fly moving upwind in this, what you'll actually encounter are periods where there's no odor or no smoke, and then all of a sudden there'll be this huge spike in odor concentration as you cross through the plume. And we can get a measurement of that in the lab by placing an odor source in front of a fan and measuring downwind with a device called a photoionization detector, which measures the odor concentration as a function of time. And what you see, if you do that, is something like this, where most of the time there's no odor, and then you see these huge spikes in odor concentration. And the signal sort of fluctuates at very high frequency. The frequency of these fluctuations and their statistics depend on exactly where you are in the plume. So if you're centered in the plume, you get these very frequent odor hits. If you move laterally, you still get these very narrow odor hits, but they're much sparser. And then the temporal scale of the whole thing depends on the wind speed. So if you decrease the speed of the fan, this whole thing gets sort of stretched out in time, and you get both longer odor encounters and longer periods between the odor. So that's one kind of signal that an olfactory system has to deal with. But there's other kinds of signals too. So odors or other substances like fog, when they're close to the ground, can form these big clouds, which some of you are familiar with, which can persist for a very long time. This can also be measured in the lab. So these are some recordings done in the Monet lab at Yale. They put a large odor source, in this case a piece of filter paper saturated with odor, on a surface. And now if you measure close to this source, what you see is a big spike initially, but then this sort of long cloud that persists for tens of seconds. So this you could think of as sort of a low-frequency odor signal, and this is a high-frequency odor signal. And this raises the question of how are odors encoded in the frequency domain? We think about this usually for the auditory system, and Frederick referred to some of this. But you could ask the same question about olfactory systems. Do they encode low-frequency fluctuations, high-frequency fluctuations? What's sort of the bandwidth of neural coding? And the reason that that's an interesting question to think about is that synapses, chemical synapses, which are the main way that neurons communicate with each other, actually behave as filters. So chemical synapses exhibit these properties which synaptic physiologists would call short-term plasticity and auditory physiologists would call filtering. So this is not really working. So I'm showing two examples of two different synapses. These, I believe, are recordings in somatosensory cortex. But synapses between particular identified types of neurons have these characteristic dynamics. So some of them depress, which means if you stimulate the synapse repeatedly, it gets weaker over time. Other synapses facilitate, which means if you stimulate it repeatedly, it gets stronger over time. And theorists appreciated right away that what this meant was that the synapse is behaving as a filter. So this is from a review by Larry Abbott and Wade Bakir, where they show 3 different types of synapses that are typical of particular neuron pairs. This one shows depression, so what this means is that it will respond transiently to a prolonged stimulus. This one shows facilitation, which means it won't be well driven by very sparse stimuli, but if you have a persistent stimulus, it will sort of ramp up and get going. And this is one that has both of these properties and therefore acts as a bandpass filter. So people have known about this for a long time, but in general it's been hard to compare what the synapses are doing in a particular area to how the neurons are responding to real stimuli, because you can't really do this in the same prep. So I don't need to sell you on the fly because Ulrike already did this, but there's lots of great things about the fly. From my point of view, one of the things that's really nice is that we can study both of these things, both the sort of sensory encoding and synaptic properties. So this is the first synaptic relay in the fly. So flies smell with their antennae. They have olfactory receptor neurons that live in the antenna and express olfactory receptors that respond to odor. They send their axons through the antennal nerve into the brain to an area called the antennal lobe, where they synapse on a number of second-order neurons called projection neurons, or PNs. So I'll use these terms, ORN, which are the peripheral neurons, and PN, which are the second-order neurons. There's also local neurons, which we call LNs, which are inhibitory, which play a role as well. So in an intact fly, we can present odors to the antenna, we can record from identified cells in the brain, and we can measure their sensory responses. We can also chop off the antenna and put a stimulating electrode on the nerve and stimulate these axons electrically and then do synaptic physiology at the same synapse. What we do if we see that is if we stimulate those axons repeatedly, we see strong synaptic depression. So we stimulate repeatedly at 10 hertz, and you can see that the response gets smaller over time. And from what I told you before, that means that the response to a sensory stimulus should be highly transient. But that's not what we saw if we presented real odors. So here I'm presenting different kinds. This is the same odor. It's an odor that I just chose because it drives a lot of cells really well. I can present a very brief pulse. Note there's a different time scale here. I can present a long pulse. This is 2 seconds. This is 20 milliseconds. And I can also present a stimulus that's sort of randomly fluctuating, as if you were flying through a plume. And what you can see both in these are individual examples. These are whole cell recordings from second-order olfactory neurons. This is membrane potential, and there's tiny little spikes. These are very typical of these cells that ride on top. Is that it does respond quickly to the onset of the stimulus, but there's also this large persistent response. And when you give a fluctuating stimulus, you get a big initial response, but it's also able to follow kind of these ongoing fluctuations really well. And that doesn't really fit with what we saw with the synapse. And we can formalize that intuition by building a model. We know a lot about the inputs here. We know how many ORNs converge on a single PN, so we can do a pretty rigorous model. And given what we know about the synapse, this is what the neuron should do. It should produce a very transient response at the onset of the stimulus, and then it should decay away. And just to compare, so in orange is the prediction of our model based on the synapse, and in black is what the neuron actually did. So we scratched our heads about this for a long time, a really long time, before we could figure out what was going on. Then we went back and looked a little bit more closely at our synaptic recordings. What we noticed is that there's actually a second component to synaptic transmission, which we'd kind of ignored because it was small and not very prominent. So you can see this big current that happens initially when there's a spike. There's also this second slow component. It's pretty small when you have an isolated spike or spikes at low frequency. But when you spike at high frequency, it actually grows. So you can see that these transient components are getting smaller and smaller, but this persistent component, the slow component, is growing bigger and bigger. So there's something else there at the synapse. And then we wonder, well, what is this slow component? And we scratched our heads for a long time about that as well. And then we sort of stumbled on something, which is that if we apply a drug, curare, so I should say these are cholinergic synapses. flies use acetylcholine as their central excitatory neurotransmitter, and they use glutamate at the muscle, which is the opposite of the way vertebrates do it. So we can apply this drug curare, which blocks nicotinic acetylcholine receptors, or at least a subset of them, at a low concentration. And that blocks the fast component, but it leaves the slow component. So then we went on a mad search through the literature to find, well, is there something that blocks the other component? And we came up with this drug, which is called imidacloprid. It's actually one of the most commonly used insecticides, and it also targets nicotinic acetylcholine receptors. And that blocks the slow component, but leaves the fast component intact. So what we think is going on is there may be two different types of receptors here at the synapse, both nicotinic acetylcholine receptors, one of which carries a fast and desensitizing current, and one of which carries a slow and facilitating current. And that allows the synapse to convey both low and high frequency signals. So here we've taken the measured properties of those two currents and built a model with two components. And so you can see that the two components together, shown in black, are able to give you a more persistent response to a long stimulus. In particular, the slow component gives you this persistent response, whereas the fast component gives you this rapid onset to a brief pulse. And this is very reminiscent of what goes on in vertebrate synapses at glutamatergic synapses, so different transmitter. But glutamatergic synapses also have two types of receptors, AMPA and NMDA receptors. We usually think of NMDA receptors as being involved in, say, learning and memory, but they also carry a different temporal content. They carry a slower component of the signal. So maybe this is sort of a general strategy for excitatory synapses to have these two different frequency components. So that's part of the answer of why we saw something different when we looked at synaptic physiology versus the intact synapse. Is there also a role for this circuitry? So I told you that there's these inhibitory interneurons that play a role at this synaptic relay. And the reason we thought about these is that previous work had shown that these inhibitory neurons actually target the presynaptic terminals of the ORNs. This was shown by Corey Root in Jing Wang's lab. So he did calcium imaging from ORN terminals, and he saw that when you applied GABA, that calcium signal decreased, showing that GABA was acting presynaptically to decrease calcium influx. And this was also shown by Sean Olson, who was a graduate student in Rachel Wilson's lab. So he showed that when you apply GABA, you can switch the synapse from showing its normal depression, which is like what I showed you, to becoming slightly facilitating. So in the presence of GABA, the initial current gets smaller, but the dynamics of the currents also change. So given this, we wondered whether inhibition was playing a role in shaping transmission at this synapse. To ask what it was doing, we simply blocked all inhibition pharmacologically and looked at what happened to the odor responses. So we saw two things. One is that the response to a brief pulse became much more prolonged. And so that means that inhibition is somehow keeping responses to brief pulses brief. However, in response to a long stimulus, normally you get this nice persistent response, but in the absence of inhibition, that response runs down over time. So inhibition is also somehow making responses to sustained stimuli more sustained. So we wondered how that could be occurring, and we wondered whether it had something to do with the dynamics of inhibition itself in the circuit. So we decided to look at the dynamics of inhibition. So to do that, first we recorded from a number of these local interneurons in response to odor stimuli. And what we observed is that although they're very diverse in their temporal responses, they tend to respond transiently. So if you look at the responses of all local interneurons, the responses are actually much more transient than those of the projection neurons. So the synapses onto the local interneurons are transient, but the synapses from the local interneurons back onto the ORN terminals also are going to play a role in their dynamics. And we found that that feedback is actually very, very slow. So this is an experiment performed by Betty Hong, who is a fellow postdoc in Rachel's lab. She expressed channel rhodopsin, which Ulrike introduced, which lets you activate neurons with light in these local interneurons. And when she activated them with light, what you can see is that the interneurons would spike right away, but the outward current measured here in the PNs took a long time to develop. It takes about 150 milliseconds for this to peak. And we can also look at presynaptic inhibition, which shows up as a decrease in these spontaneous events. So here we're measuring the standard deviation of the PN current as a measure of these spontaneous events. And that also decreases slowly, again, taking about 150 milliseconds to peak. So inhibition is transient, it's slow, and it's presynaptic. Can these properties together account for the observed effects of blocking inhibition? And it can. So here I'm showing you, on the top row, I'm showing you data. So this is what the neuron does both in the intact circuit in black and without inhibition in purple. And here's our model. So if we just put those two components, the fast and slow components of synaptic transmission together, you get the purple trace, which runs down over time. And inhibition both makes the response to a brief pulse brief, and it stabilizes the response to a long pulse. Now, because this is a model, we can play games with this and say, well, what if it wasn't transient, slow, and presynaptic? What would happen? So is it important that inhibition be transient? Well, we can ask that by replacing transient inhibition in our model with inhibition that turns on and stays on. And if inhibition turned on and stayed on, then the response of the neuron would run down over time, just like the synapse does. So it's important that it be transient. Is it important that inhibition be slow? We can ask that by speeding up the time course of inhibition. And if inhibition came on more quickly, then it would hit as the response of the projection neuron is still rising and slow it down. So inhibition needs to be slow in order to preserve the rapid transmission across the synapse. And what if it were not presynaptic? So we can change our model from a presynaptic inhibition model to a postsynaptic inhibition model. And one thing we see with presynaptic inhibition is that having inhibition actually increases sort of the signal-to-noise ratio, so that is the difference between the odor-evoked signal and the baseline. And that's because presynaptic inhibition tends to have more of an effect at low firing rates than at high firing rates. I can tell you why that is if you're interested. In contrast, postsynaptic inhibition does have more of an effect when the cell is depolarized. That's when it's further from the reversal potential for inhibition. And so if inhibition were postsynaptic, it would decrease the signal-to-noise ratio instead of increasing it. So finally, because this is a model, we can now play other stimuli that are hard to generate in the lab, like sinusoids, and this can give us a picture of what inhibition is doing functionally. So if we present sinusoids at different frequencies, we can look at the response both with inhibition and without. And what you can see is that without inhibition, this synapse acts as a bandpass filter. And what inhibition is doing is sort of flattening this response, so that it's allowing the synapse to transmit signals more reliably across a wide range of frequencies. So what I've shown you is that there are multiple mechanisms present in the circuit that promote broadband encoding of odor stimuli. First, there's two components of transmission at the feedforward synapse, and then there is this dynamic inhibitory loop that sort of flattens the frequency response. And I just want to close with the thought that this idea that really came from Allison, that we need to think about the dynamics of natural stimuli when approaching a circuit, led us to discover many new things about the biology. We discovered a novel component of synaptic transmission that had not been described here before, and a novel role for inhibition in this circuit. I've talked about this a number of times, and one of the questions that I'm always asked is, why do we actually need multiple mechanisms here? Why not just build a synapse that can transmit all frequencies? I don't know the answer to that, but I want to leave you with an idea and a metaphor, which is that we could ask the same thing of people who build stereo systems, that I'm showing here. And why not just build a stereo system that can, that has great bass and also great, really clear treble? And the auditory engineer would tell you, well, that's really hard to build. And in fact, the two of the tricks that they use, the auditory engineer uses, are two of the tricks that I've told you here. One is to split things up into different frequencies. So you use one speaker for the bass, one speaker for the middle range, one speaker for high frequencies. And another is to use feedback to cancel out, to remove sort of distortions in the signal. So that's one possible answer, is that it's actually difficult to build a broadband synapse, and so you have to build it out of these components. A second possibility is that just like in an auditory system where you have an equalizer, sometimes you want to hear more bass, or sometimes you want to hear, really bring out those high notes. Maybe the fly has to do the same thing. So if it's flying through the air, it may want to bring out the high frequency fluctuations. If it's moving along the ground, it may want to adjust and bring out the low frequency fluctuations. And having a circuit that has these sort of moving parts may allow it to adjust what it transmits to match the environment better. So Allison was always, she was a scholar, and she was meticulous about assigning credit to people. When she gave, I remember very distinctly her giving a talk about my work, and like most people who would say, we did this, we did this, we did this, here's the person who did the work. She would say, Kathy did this, and Kathy did that. Mimi did this, Mimi did that. So I want to end by saying thank you. This work was all done in the lab of Rachel Wilson, and in collaboration with my wonderful colleague Betty Hong, who did the channel rhodopsin experiment. It was inspired by the work of another postdoc in the lab, Emery Yaxie, who did some of the first recordings of Ellen's that inspired this project. And I got invaluable feedback on this project from Gabe Murphy and from my husband, David Chopek, who's somewhere around here with the baby, who's been a collaborator and scientific partner for many years. And this project was really, I think, inspired by Allison in more ways than I can recount, from the ideas that drove it, the approach that I took to how I presented it. I feel at my thesis defense, Allison called herself my Dr. Mutter, my scientific mother, and that's how I think of her. I wouldn't be in science today if it weren't for the influence that she had. Those who knew me as a graduate student know that I didn't come into graduate school with the thickest skin. Having a thick skin is a useful skill in science. And she was the perfect advisor for me, because I would give a talk, people would ask hard questions, and I would go back and I would say, I am so depressed, I feel terrible. And she would say, I know, I understand, it's so hard. Get back out there. So I really owe the confidence that I have as a scientist to her. And I feel lucky that I was able to come back to UCSF and give lab meeting in her lab last year and talk about a little bit of this work. And I really wanted to come and tell her, this is what I did with what you gave me. And I wanted to tell her thank you. But I couldn't say it at the time, because if I had said thank you, that would have meant saying goodbye. So thank you.